Even when you think you know quite a bit about something, there's always more to learn. Well, hello and welcome to another episode of You and Me and Multiple Sclerosis. My name is Pam. I've been living with multiple sclerosis for 40 years as of January 5th, 2025, which was a couple of days before I posted this video. And I've been in a number of MRIs and I've seen a number of brain scans, my own and others, and I thought I kind of understood all about lesions and what they signified, and even sometimes where they're located and how that might affect certain symptoms that I have. But, in looking at the research that's still going on, I'm finding there's more to learn about lesions. But before I get into the articles that I wanted to share today, I found an interesting little video that I thought would be good to share with you. I was looking for a good photograph or some kind of illustration that would just show what lesions look like on a brain scan, just for those of us who either haven't seen it or just want to be sure we're all talking about the same thing. Well, I found this, and it's pretty cool. It's a time-lapse MRI from a patient with MS. And it shows both an expanding rim lesion and a shrinking lesion. Let's take a look. Because you can see that that one down with the red box, you can see how over time things seem to bloom and fade, but overall that it is expanding. Well, that was different. I was glad to have run across that because seeing it in a time lapse actually is much more of an illustration of what lesions do than just seeing the still photos that we get with our MRI scans. Let's go ahead then and look at the couple of papers that I found. One of them, the first one we're going to look at, talks about a way of predicting cognitive decline based on the number of lesions a person has. The first article I saw was in MedPage Today called Cognitive Decline in MS Predicted by Paramagnetic Rim Lesions. And the subtitle says Processing Speed Scores Lower 4 years later for people with one or more baseline lesions. This was a pretty recent publication. It came out on March 4th, 2024. And as they say here, paramagnetic rim lesions, or PRLs, predicted cognitive decline in multiple sclerosis data from a longitudinal cohort study. The article goes on to say that at year 4, scores on the single-digit Modalities test, a measure of cognitive processing speed, were lower for MS patients with at least one baseline PRL compared with those who had no baseline PRLs, reported Hannah Schwartz, a clinical research coordinator at Weill Cornell Medicine in New York City, during the America's Committee for Treatment and Research in Multiple Sclerosis, or the ACTRIMS forum. And I think I've had that test. Isn't that the one where, yes, you're supposed to look at different symbols and then put the number down that corresponds with them? You're told, here's the key up to at the top of the test, but it's really measuring your ability to remember and react quickly. And so they're saying is that the more of these lesions you have, the lower your score tends to be. And they go on to say that in a multivariate model, people with at least one PRL at baseline had average adjusted SDMT scores that were 4.1 points lower at year 4 than patients without a PRL. The SDMT is a 90-second time test. Scores range from 0 to 110, with higher scores indicating faster processing. And quite frankly, I have no idea how I did on those tests. Paramagnetic lesions, they say, may identify patients who are at risk of a more aggressive disease course, specifically in terms of cognitive decline, Schwartz said. There is considerable research around developing prognostic biomarkers for MS, and our results are a first step toward the possibility of applying these PRLs in a clinical setting. While cross-sectional studies have demonstrated an association between PRLs and worse cognitive functioning, there's limited data about future cognitive impairment, she added. MRI traditionally has been used to detect new lesions in MS, noted co-authors Susan Gauthier, DO, MPH, also of Weill Cornell Medicine. As a result, chronic lesions or scars are often overlooked. However, recent studies have shown that some chronic lesions exhibit ongoing inflammation at their rims, indicating persistent immune activity that could cause further damage, Gauthier told MedPage Today. Paramagnetic rim lesions, a type of chronic active lesion, contain iron within the immune cells at their rim, making them detectable via MRI. Our study adds to evidence that paramagnetic rim lesions are associated with a more aggressive disease progression, she added. With MRI's ability to identify paramagnetic rim lesions, there's a concerted effort to leverage this imaging biomarker in clinical practice, potentially revolutionizing the use of MRI for patient care. The study included 106 MS patients with MRI and cognitive data from an ongoing longitudinal study. Schwartz and co-authors used quantitative susceptibility mapping, a post-processing imaging technique, to identify PRLs on imaging. Makes me wonder if it's post-processing. I'm kind of wondering, could you go back to people's old MRIs now that we have this new way of thinking about it? Could you look at somebody's old MRI and see perhaps what their baseline might be? Interesting idea. Other imaging variables included T2 fluid attenuated inversion recovery, or FLAIR, total lesion volume, cortical thickness, lesion myelin water fraction, and thalamic volume. Baseline and 4-year cognitive evaluations were performed using the Brief International Cognitive Assessment for Multiple Sclerosis, or BICAMS, which includes the SDMT, California Verbal Learning Test 2, and the Brief Visual-Spatial Memory Test revised. Univariate linear models assess the relationship between baseline imaging features and cognitive function at year 4. Multiple linear models were performed with all possible variables, and final models were determined based on significant variables only. Of the 106 participants, 73.6% were female. Mean baseline age was about 43. Nearly all patients, or 92.5%, had relapsing remitting MS, and most, 69.8%, were on highly effective disease-modifying treatment. At baseline, 38.7% had at least one PRL, and the mean expanded disability status score was 1.0, indicating minimal disability. They say that patients with PRLs performed worse on all cognitive tests, Schwartz said. In the univariate model, the gap between PRL and no PRL groups widened at 4 years, although only SDMT reached significance. In a separate multivariate model, BVMT scores at year 4 were associated with the presence of PRLs when controlling for baseline PVMT and disease duration. Participants with at least one PRL at baseline had an average BVMT score that was 1.9 points lower at year 4 than patients without a PRL. There was no relationship between PRLs and CVLT at year 4. I think if you're a statistician, all that makes sense to you, but I'm kind of just gathering that it must be somewhat statistically significant, or the findings of the research would not really have been worth reporting on. They go on to say, other MRI variables tied to subsequent cognitive function included overall lesion myelin damage, which was linked with SDMT, and total lesion volume, which was associated with CVLT. The study had several limitations, Schwartz acknowledged. The cohort was small, and imaging data were available only at baseline. BICAMS is a limited cognitive assessment, she added. We would want large multi-center studies to replicate this result to evaluate the full potential of PRLs as an imaging biomarker to identify patients at risk for cognitive decline, she said. We might be able to treat them more aggressively to preserve their cognition. And I think that would actually be helpful. I know that nowadays many doctors are suggesting to their patients to begin with the most aggressive treatment available right away because you're more likely to nip your MS in the bud that way. Although it's certainly not 100% of all doctors who have that line of thinking. But if you think about people who might be hesitant to go on a really aggressive DMT if they aren't even all that disabled yet, and I think I probably would have been in that category myself, knowing that you are at risk of cognitive decline could inform your decision and make it more likely that you will go on a more aggressive DMT in the hopes that you're going to stave off cognitive decline. And I'm thinking just for myself, I think that would probably have pushed me over to where I would have said, yeah, sure, I'm going to try it because I want to hang on to as much of my cognitive function as I possibly can. And then the second paper is going to talk about where lesions might actually originate, which I guess, now that I think about it, is a mystery. I didn't know, and I never heard anyone really say. It's just lesions are there, but apparently there are reasons why they happen, and this paper purports to be addressing that topic. Once again, as I'm seeing more and more with the research that we're looking at, a lot of times it seems like it's not so much that the body is misbehaving with any kind of intent. So it's really kind of unfortunate that the medical community refers to these things as attacking, like the immune system attacking itself, which sounds so violent and purposeful. But more that maybe the right thing is in the wrong place or is mistaken about what it's seeing and what it's supposed to be doing. And I think that you'll get that idea from the second paper. This one I saw in Multiple Sclerosis News today. It's called MS Lesions May Start as Small Clumps of Microglia in Patient's Brain. Tissue samples with these nodules also show more lesions than active lesions. And this was published March 12th of 2024. And they say that in multiple sclerosis, lesions in the brain may start with small clusters of immune cells called microglia, a new study reveals. And here's what an artist thinks that looks like. The microglia are in orange. They look kind of menacing, don't they? Scientists are working to understand exactly how these small clusters may develop into MS lesions, which they hope could uncover new targets for treating the disease. Now we need to explore the relationship between all these inflammatory components, so we can understand exactly what leads to these early signs of breakdown. After that, we can start thinking about which steps we can remove from this process to avoid the development of new lesions altogether. Alida van den Bosch, a neuroimmunology group researcher and study co-author at the Netherlands Institute for Neuroscience, said in an Institute press release. The study, Profiling of Microglia Nodules in Multiple Sclerosis, reveals propensity for lesion formation, was published in Nature Communications. What causes MS lesions to form is not fully understood. It should say fully understood. I guess they needed a proofreader and they did not get one. Anyway, in MS, inflammation in the brain and spinal cord leads to regions of damaged, scarred nerve tissue. These lesions are a hallmark of the disease, but how they develop isn't fully understood. Microglia are resident immune cells of the brain. They help to protect the brain against infection and also act as the brain's main sanitation workers, helping to collect and dispose of molecular debris. So once again, all of these things in our brains and bodies were designed for a good purpose. And when they're functioning as they should, and when they're located in the place that they should be located, they do a wonderful job. It's when they either get into the wrong place or they get confused in some way they start doing things that they shouldn't do, and that's why we end up with various diseases, including multiple sclerosis. And they go on to say it's been known for decades that microglia in MS patients sometimes form atypical clumps called microglia nodules. These nodules are seen in brain tissue that otherwise doesn't appear damaged, and some researchers have proposed they could be an early sign of lesion formation. The biggest problem with that theory is that microglia nodules aren't just found in MS. They're also seen in other neurological disorders like stroke, but those conditions are not marked by lesions. So if microglia nodules are indeed the early stage of MS lesion formation, there must be something about these nodules in MS that differs from other disorders. To learn more about the role of microglia nodules, scientists analyzed brain samples from the Netherlands Brain Bank MS cohort. In samples of post-mortem brain tissue from 167 MS patients, about 2/3 or 64% had detectable microglia nodules. These patients also had more lesions and more signs of active inflammation in the brain compared with samples from patients with no detectable nodules. We found that patients with these nodules have a worsened pathology. They have more lesions, and the lesions are more active, Van den Bosch said. Microglia nodules also in brain tissue affected by stroke, but with differences. Scientists then compared microglia nodules in brain tissue from people with MS to those in tissue samples from people who had a stroke. As microglia nodules in stroke are not involved in lesion formation, differences between microglia nodules in MS and stroke may reveal mechanisms beyond MS lesion formation, the scientists wrote. Results showed microglia nodules occurred more frequently in MS brains, but the nodules were of comparable size in both disorders. Additionally, some microglia nodules in MS brains expressed many inflammatory genes that are known to be expressed highly in MS lesions, and these inflammatory genes were not expressed highly by microglia nodules in stroke brains. We recognize that many of the genes in the MS nodules quite quickly because they're also expressed in the active lesions, Van den Bosch said. Nodules in MS brain tissues, but not of those in stroke, also showed signs of a generally more inflamed environment, such as higher counts of nearby inflammatory immune cells. MS is marked by damage to the myelin sheath, which is a fatty covering that surrounds nerve fibers. The researchers found that nearly half of microglia nodules in MS brains, but none in stroke brains, were in close proximity to nerve fibers that had damaged myelin. These microglia nodules also express genes that are known to be important for breaking down and processing fats like those found in myelin. When we looked at the axons or nerve fibers surrounded by the nodules at high resolution, we saw that they were associated with partially demyelinated axons. These nodules likely arose to clean up the oxidized or damaged myelin, Van den Bosch said. According to Van den Bosch, microglia that become activated both by the metabolic changes needed to break down myelin and inflammatory signals from other immune cells may become very inflammatory, causing more damage to the tissue surrounding them, leading to a sort of downward spiral that could ultimately lead to the formation of MS lesions. Study results suggest that some microglia nodules in MS are likely sites of lesion initiation and represent an interesting therapeutic target to prevent early demyelination and MS lesion formation, the researchers concluded. Well, okay, so now we know more about lesions. Unlike some of my videos, this is not one where you can say, okay, from now on I'm going to do this, or I'm going to stop doing that, or even what questions might I ask my doctor so that I'm more informed. But at any rate, I thought this was interesting just to know basically that there's some things going on. The second paper, I think, is in the realm of fundamental research. And remember, I've talked about that before. Fundamental research is trying to understand basics rather than applied research that says, okay, now that we know all this stuff, how can we apply it to a problem? I don't think the microglia hypothesis is ready for prime time yet, and even if it is, I don't think it's translated into a treatment strategy, but it could become a building block toward that. And the first one about cognitive decline and the presence of lesions, well, that one got my attention. It probably got yours too. It's interesting, again, to know this stuff, even if it's not going to change anything about how we live our day-to-day lives, except to say that now you understand why keeping your general health just as strong as it possibly can be is going to benefit you. No matter what MS throws at you, you will be as ready for that as it's possible to be. So take good care of yourself, and I will see you again in my next video.